see. There we go. So in this video, we're going to continue to look at proteins that are associated with the membrane, and we're going to look at the lipid modifications that can link these proteins to the bilayer. So in the previous video, we looked at the proteins that are integral. Here we're looking at proteins that are just attached via a lipid. So one type are proteins that are called prenylated. And these are covalently attached to lipids that are built from isoprene units. And so here are the two types, our common types, are farnesyl and gurnal lipids. So you can see the isoprenyl, if you remember that from a couple videos ago. So you might want to review and see what that looks like. But you can see it's just a repeating over and over and over again. To attach these proteins, they're usually attached at a site that is cis XXY. Your X is an aliphatic amino acid, and your Y is either going to be almet or SER for farnesyl. And if it's a Y that is leucine, then it's gurnal. And the way that you attach these lipids is through the sulfur of cysteine. So here we have that lipid attachment. This lipid can go into the membrane, and now you have a protein that's part of the membrane. These type of lipid modified proteins are usually on the cytoplasmic face of the plasma membrane. So they're facing inwards towards the cell. Another type of modification you can have is myestrylation and palmylation. So mystrytic are rare type of modifications. Here's mystrytic acid, and here's palmitic acid. So that's more common. And so myestrylation happens at N-terminal glycine residues. So you have a glycine at your N-terminal, you can add this fatty acid to it. And once you add it, it's going to be there until the protein is destroyed. It is stable for the protein's entire lifetime. These type of modifications are in your cytosol, your ER, in the interface again of the plasma membrane, so facing towards the cell. And you will also find these modifications inside the nucleus. Palmitylation, this is at cis residues, and this forms a thioester bond. And again, this is the cytoplasmic face of the plasma membrane, and this is used in signaling. So these proteins that have the palmitic acid, they can send signals across the membrane. And the way that you remove this doesn't attach for the cell's entire lifetime, is through palmitoyl thioesterases. So you can cut your palmitic and your protein linkage, and your protein can go free because it's no longer bound to the membrane. Now you can also have glycosyl phosphatidylinositol linked proteins, which is easier to say as GPI. And so you have this phosphatidylinositol as part of your head group, and then you have a bunch of sugars, then the phosphatidylethylamine attached to your protein. So you can see your protein has this giant thing attached to it. Here, GPI proteins are actually on the outside of your plasma membrane. So these are not on the cytosol side like we saw in the previous slide, but they're outside facing. And the type of linkage is uniform. You have this phosphatidylinositol, 3 mannoses, and 1 glycoamino residue connected to your inositol. And your mannose will form a phosphodiester bond with the phospholinositolamine as we see here, and then you attach it to your protein. And again, these are anchors. This will help to anchor your protein into the plasma membrane. Now, we have talked about integral membrane proteins, which have to be part of the membrane, but peripheral membrane proteins, as shown here, they can actually dissociate, so they don't have to be part of the membrane. And unlike lipid anchor proteins, they do not actually bind lipids. They're just kind of shoved in there, and they can pop right out. And what happens when you remove them from the membrane, it's like they're soluble proteins. So they're a very interesting class of proteins, to say the least. That's it for this video. I will see you in the next one.